The coronavirus pandemic, which began in late 2019, has changed all of our lives. It's affected the world in a million ways, but some places have stayed the same, like the Centre for Virus Research. The University of Glasgow's Centre for Virus Research was established in 2010 in partnership with the Medical Research Council. This centre contributes vital research on viruses and has played a critical role in the global response to the coronavirus pandemic. The centre was named as one of the 13 key centres in a UK alliance of scientists working on coronavirus whole genome sequencing. Today I'll be talking to Dr. Anna Filipe, who manages the next generation sequencing facilities at the centre. Before we hear from Anna, I thought I'd give you some context about what sequencing is. But don't worry, Anna explains it for us later if you still aren't sure. Sequencing is a way of reading DNA or RNA genomes. In simple terms, a genome is a set of instructions that is needed to build something, whether that's a human or a virus. Genomes are very long lists made up of a combination of only 4 letters: A, C, G, and T. This technology lets us read these letters in the correct order, which lets us understand more about whatever we are sequencing.
So my name is Anna, and I've been working at the CVR over the last 12 years already. Currently I head the genomics team. What we do in our group is really to support different projects at the CVR whenever they need any type of help with high-throughput sequencing. Well, on one hand, I really enjoy new technologies. I think it's really exciting, but genomics is amazing because of the amount of information that you can extract from nucleic acid sequences from genomes. And we can do things as diverse as understand drug resistance, tracking evolution of viruses, trying to understand based on genomes if there is a higher association, for example, with disease severity or escape to treatment. So it's really the high diversity of responses that you can have simply by looking at genomes. I initially trained as a pharmacist, but I also had a chance to do an internship in research, specifically in gene therapy. And I really fell in love with research by then, so I decided to do a PhD in that area. As part of that PhD, I also had the chance to explore viral vectors as tools to treat diseases. And that really was the link to this center of virology. So I moved here to start a postdoc in virology. Specifically, I was developing assays to test inhibitors of hepatitis C virus. And soon after, I just happened to get the chance to work on high-throughput sequencing to help identify mutations associated to drug resistance. And a couple of years after, there was this opportunity to lead the genomics team at the CVR, so I took it, and that's what I've been doing since then. I don't think my job is as any type of routine, really. It's quite diverse, but as we are a group whose main purpose is to support other research projects, a lot of what I do is really plan and discuss results associated to those projects that we support. I also have to do a lot of prioritization work, and also very important is always to keep an eye on the new technologies and then propose them for implementation, and also strategic planning at the institute level and also in the group. So in order to explain next generation sequencing, we first need to understand what is a genome. And a genome, in a simplified way, is a sequence of 4 letters, A, T, C, G, that we call nucleotides. And it's the way all those nucleotides are organized that really dictates the storage of information and all the phenotypes that we see around us. So sequencing is nothing more than a group of technologies that allow us to read those genomes. And those automated genome readers are called sequencers. Once we have produced those bits of information, then that data really needs to be reassembled back to be able to rebuild those sequences and the original genome. Really, the big revolution with next generation sequencing had to do with a decrease of cost of the process and also the amount of information produced and the speed at which it can be produced. Because with next generation sequencing technologies, you can have billions of sequencing reactions happening in a single chip. So just to give you an idea, the Human Genome Project, which started in the 1990s, was aiming to get the human genome sequences available over a period of 15 years. They did it faster than that, but nowadays we could sequence a human genome in less than 2 days. Overall, there are still very high upfront costs associated to purchasing equipment, maintaining it, all the infrastructures. They are not very accessible. And I think one of the downsides of it is that countries that have more access to resources do have a higher sequencing capacity as opposed to other countries that don't manage, for example, to access this type of technology. So next generation sequencing has really permeated across so many disciplines, not only in the area of research but also in the clinical arena. And it has been used, for example, for whole genome sequencing, including identifying variants, and this has been really important in terms of disease prognosis. A different application is called Metagenomics. That's when you try to sequence everything that exists in your sample, and it can be used to identify communities of bacteria, of viruses that might exist in a particular sample. So if you don't know what is causing a disease, that is a good way to approach in terms of high throughput sequencing. There are several other techniques, sequencing techniques, that can also help you to understand how does the viral infection affects the host homeostasis. So you can do what we call RNA-seq, for example, where we can see all the transcripts that are being produced by a host when it is infected. We can look at protein binding sites, for example, or even look at epigenetic markers in the sequences and how they are controlling the expression of transcripts of genes. As viruses make copies of themselves as they multiply, there are errors that are introduced in their genomes, and therefore through sequencing we can actually read those alterations and be able to track them back and study how the virus is transmitting and how it is evolving. It really depends on the virus and depends on the diversity associated to the virus. So I can give you an example with SARS-CoV-2. When it first emerged in the human population, there were hardly any changes being introduced in the genome. So it was really difficult. We were just producing sequencing data, but everything was very similar. So it's very difficult to track the virus track introductions and transmission networks when a virus is really not changing that much. It's also important in the context of evolution of understanding what is it that is causing those changes. Is it just random mutations, or is the virus under some type of pressure from the host, from vaccines, that is actually affecting the mutations that are being introduced in the genome? So another important aspect whenever we are looking at virus evolution is that it really depends on the global scale of the problem. For example, we have been doing loads of sequencing for SARS-CoV-2 in the UK, but there are many regions of the globe where the sequencing coverage is very, very low. So whenever you see a new variant emerging, it's really difficult to understand how it has evolved because of the lack of information in other geographical areas. And also, it's very, very difficult to really get clear answers sometimes when a virus, for example, jumps from an animal to the human population. When did that take place? And getting those dates accurately can be really, really tricky. So as with any other technique, we really need to be careful with the limitations of sequencing, and there's definitely errors that can be introduced during the sequencing process depending on the library preparation methodology that was used, depending on if there were loads of PCR cycles done or not. So we need to be very, very careful with the interpretation of the data. And in order to help us deal with that, there are many controls that we have to introduce, not only generic control measures about where we do each one of the steps to prevent contaminations, but also include controls in our sequencing runs to help us have be assured with the results that we get. So that has actually to do with the technology that are used to sequence the virus. So SARS-CoV-2, whenever we use clinical samples as our source material, the virus exists in very, very low amounts. So we need to find ways of specifically amplifying the genome of the virus before we do sequencing. In order to do that, we need to use primers that have been designed specifically for known viral sequences. So what happens as the virus evolves and mutates is that those primers that initially were developed might not work as well. So this is something that needs to be kept under monitoring whenever we are doing the sequencing of SARS-CoV-2. Both influenza virus and coronaviruses, they are types of RNA viruses, and RNA viruses are known to have a higher rate of mutation when compared to DNA viruses. However, coronaviruses have a large RNA genome, and therefore they have this enzyme called an exonuclease that brings this level of proofreading when the virus is replicating, and therefore it has a lower level of mutations when compared to influenza. It's really having access to such a widespread diversity of expertise. You have colleagues with such a diverse range of backgrounds, and the fact that they are all here available for a corridor chat if necessary is really exciting. It's quite impressive because when I started doing this job in 2016, I was really wondering how much would other PIs, other investigators at the CVR warm up to the idea of using NGS as a tool, and definitely the interest has been quite impressive. But also when you look at the market projections for next generation sequencing, it's definitely not going to reach a plateau anytime soon.
Woo!